Hello to all my dear lovely students. I hope you all are doing fine. So once again, I welcome back you on this amazing platform of Physics Wallah, where we are studying in excellence batch. So far we have already completed chapter 1 and now we are going to start with the chapter number 2, that is sexual reproduction in flowering plants. I hope you all have gone through all the lectures of chapter 1, that is reproduction in plants, which is general chapter. Now you're going to see that how sexual reproduction takes place specifically in plants. Yes or no? Okay. Now beta, in your last lecture you have seen the events of sexual reproduction, that first gametes need to be produced, followed by their transmission. Then there is fertilization, and then there is zygote formation, followed by embryogenesis. So in your last lecture you have only seen the headings, that okay, gamete has to be formed, okay, their transmission has to be done. Now in this chapter you are going to study all those things in detail, that how a higher plant, angiosperms, flowering plants, actually produce gametes, transfer them, fertilize them. Everything you are going to study in detail in this chapter. Correct? Very good. So today we are just starting the chapter, that is sexual reproduction. So today we are going to first start with the sex organ of the plant body itself, which is flower. Whatever this process, whatever sexual reproduction takes place in higher plants like angiosperms, the specific organ is created for that, which is flower. So today first we are going to study about what is flower, structure of flower, and then we are going to study the details of gamete formation, their transmission, fertilization, and so on. Right? Chalo. So let's start with today's lecture without wasting time. So the first topic in the class says introduction, that is structure of a flower, where actually you have to perform sexual reproduction is actually flower. So first we are going to study about flower, and then we are going to start with the main process. Now beta, in class 11th there is one chapter by the name morphology. In morphology you already study this topic, but let's do a quick revision. Yes or no? So this is going to help you. This quick revision is surely going to help you to understand that what is a structure of flower and where actually this sexual reproduction takes place. So if I talk about flower, which is the sex organ of the plant, let me write this first. It is sex organ. It can also be defined as the modified shoot. How can you define flower? Flower can also be defined as modified shoot or super specialized leaf. So there are three ways to define flower. Super specialized leaf. Now, okay, it is a sex organ. This though we can understand, because here sexual reproduction takes place. Why it is called as modified shoot? So beta, flower is known as modified shoot because the shoot apical meristem transforms into floral apical meristem. Shoot apical meristem modifies into floral apical meristem to produce flower with the help of hormone florigen. So that is why it is known as modified shoot, because to produce flower, first shoot has to get modified into floral apical meristem. Understood? Now, why it is known as super specialized leaf? It is known as super specialized leaf because from where actually originally leaves and branches would have arisen, but due to the modification of the shoot, now from there flower will arise. So we can say that flowers are super specialized leaves which are actually responsible for showing sexual reproduction. So I hope all the three terminologies related to flower is clear. Yes? Okay. Now structurally beta, flower can be divided into 4 whorls, and these 4 whorls can be kept under 2 headings. So structurally flower is having 4 whorls. These 4 whorls can be kept under 2 broad headings as non-essential whorls and essential whorls. I'll tell you what is this with the help of the diagram. Right? So how many whorls are there? Total 4, divided into non-essential and essential. In non-essential beta, there are 2 whorls. The 2 outer whorls in non-essential category are calyx and corolla. Whereas in essential whorls, the 2 which comes are Androecium and Gynoecium. Correct? Yes or no? Now, how many whorls can you see? 4 whorls. The outer one is calyx. Inner to calyx is corolla. But a calyx represents group of sepals. Group of sepals are actually calyx. Followed by calyx are group of petals. The colored structure which you see, with which you get attracted, is actually the petals, and collective term for petals is corolla. Right? These 2 are non-essential whorls because they do not take part in sexual reproduction directly. They are indirectly involved in this process but not directly. Here there is no gamete formation or transmission taking place. So therefore they are known as non-essential, as they do not participate in sexual reproduction directly. Correct? Yes or no? Now, inner to calyx, inner to corolla is the third whorl, which is known as Androecium. Androecium represents the male reproductive organ, and it is a collective term for stamens. Right? It represents the male reproductive organ. The innermost whorl after Androecium is Gynoecium, also called as pistil, right? And it represents the female reproductive organ, collective term for carpel. So I hope it is understood. The first 2 are non-essential whorls, followed by 2 essential whorls. Now why are these essential? Because these 2 whorls, Androecium and Gynoecium, are actually responsible for the production of gametes. They take part in sexual reproduction directly. Androecium is responsible for the production of male gamete, whereas Gynoecium, carpels, are responsible for the production of female gamete. So therefore they are essential. The other 2, the outer 2, are non-essential. Correct? Now let's see with the help of a diagram. Beta, all these 4 whorls, they are arranged on a swollen structure called thalamus. So this is a thalamus. So this is what? This is a swollen base on which all the 4 whorls of the flower are arranged. The swollen base is known as thalamus. Can also be called as torus. Can also be called as receptacle. Right? On this swollen base is arranged all the 4 whorls, starting with the outermost non-essential, which is known as calyx or group of sepals. So these are what? These are actually sepals. The number of sepals varies from species to species. Right? Now, inner to sepals are group of petals, also called as corolla, which are actually non-essential whorls, attractive, colorful. Therefore, this is how you have to draw the petals. So what this represents? These represents your second whorl, that is corolla. Right? These 2 are non-essential. I hope you have understood this. Moving ahead to the third whorl from outer to inner region is Androecium, commonly called as stamens, which represents the male sex organ. I'll explain you in detail. So this is stamen. Inner to stamen, the innermost whorl is your the female reproductive organ, which is responsible for producing female gamete called carpel. Right? So these are the four whorls: number 1, number 2, number 3 and number 4. Which all four whorls, they are born on swollen base that is called as thalamus. I hope the structure, this simple structure of flower is clear. Details we are not going to do over here because you have already done in class 11th morphology chapter. Now beta, in this chapter we are actually going to deal in with stamen and pistil. We are going to deal in with Androecium and Gynoecium. We are not going to deal in with calyx and corolla because we want to study that how in plants gametes are produced. Because we are studying about sexual reproduction, and the first event in sexual reproduction is to study gamete formation. So first we are going to learn that how male gametes are produced in plant on this special structure called stamen, and then we are going to study that how female gametes are produced on the special structure called carpel. So now let's start the structure of Androecium in detail, right? First we are going to study the formation of male gamete, and then we will study about formation of female gamete. Chalo, see, structure of Androecium. First of all, you should know what is Androecium. Androecium is a collective term for stamens. It represents the male reproductive organ, right? Which is found inner to corolla, and outer to pistil, right? So this is the location of Androecium in the flower. I hope you all have understood this. Now let's try to understand the structure of a typical stamen. Number of stamens together, they are called as Androecium. Now see that how the stamen looks like and where in the stamens actually male gametes are produced. So structurally, stamen consists of 3 parts. Typical stamen is made up of 3 parts: anther, filament and connective. The 3 parts are anther, filament and connective. So first I am going to draw all the 3 parts and then we will discuss each part in detail. So this is my swollen base and this is the stamen attached to this swollen base, right? This is whole stamen. The above swollen part which you see is known as anther. There are usually 2 lobes of anther which are joined with the help of connective. Right? And the stalk which you see with which the lobes of anther are attached to thalamus, right? Or petals. Petals are also there over here only, na? These are the petals. So this stalk is known as filament. Understood? Yes or no? Now beta, there are 2 ends of filament. The upper end which is known as distal end and the lower end which is known as proximal end. So this is the distal end of filament, whereas the lower is the proximal end of filament. Now tell me, the distal end of filament is attached to anther. So what will you write? What is filament? In filament, what are you going to write? The first point, Filament is a stalk. It is a stalk which connects anther lobes to the thalamus, right? Now, the distal end of the filament is attached to this question is usually asked in NEET because it is given in NCERT that the distal end of filament is attached to the lobes of anther, whereas the proximal end is attached to either thalamus or petals. Can you analyze this with the help of the diagram? Shall I write these 2 points? Yes, ma'am. Chalo, write it. So what is filament? First of all, filament is a stalk of stamen, right? Second point, whose distal end is attached to, anther, very good, and whose proximal ends are attached to either petals or thalamus. You have seen in the diagram, either petals or thalamus. Correct? Any doubts to anybody? Now, next portion after filament in a stamen is anther, which is very, very important. Actually, in anther only male gametes are produced. Male gamete carrying body called pollen grains are produced. So, anther is the main sexual part of the stamen. We say that stamen is a reproductive organ. But specifically in stamen, which is the reproductive organ? It is the anther, because the male gamete carrying body, pollen grains, are produced in anther. Correct? Usually in 95% of the angiospermic plants, there are 2 lobes of anther which are joined with the help of a sterile tissue called connective. Right? Now, if you look into each lobe of anther, so each lobe of anther, they have 2 chambers. So each lobe is bithecous. There are 2 chambers means there are 2 thecas. So each lobe is having 2 thecas, chambers, in which actually pollen grains are produced. The male gamete carrying body, pollen grains are produced, right? So what we say that first of all, anther is the site of pollen grain formation. Pollen grain formation. What is pollen grain, beta? It is actually a male gamete carrying body. Right? It is a male gamete carrying body. Keep it in a bracket so that you understand it. Okay. Second point. Anther, typical anther in 95% of the angiospermic family with few exceptions are bilobed. Bilobed. Can you see there are 2 lobes? These are the lobes of anther. Each lobe of anther have 2 chambers, have 2 thecas for the production of pollen grains, for pollen grain formation. Correct? Yes or no? So what term do we use for this big statement? Dithecous or bithecous, right? So what term are we going to write over here? Dithecous. Hence we say that our anther, typical anther of angiospermic family is bilobed and dithecous. Bilobed, 2 lobes, and each lobe has 2 chambers. So we can say thus anther is bilobed and Dithecus. Correct? Any confusion to anybody over here? No? No, ma'am. Very good. So now let's proceed further. Now beta, exceptions are there, but exceptions we need not to do. One exception is like in Malvaceae family, that is your ladyfinger, China rose, cotton family, where anther instead of bilobed bithecus, it is monolobed bithecus. But we are not going to do it, so just leave that portion. You remember the general structure of anther. Now if I want to draw anther separately, then I will see that anther is bilobed bithecus and has 4 angles. They have 4 sides. Now see, this is how I am going to draw anther. I don't have much space, so I am going to draw anther separately over here. Later I'll draw it in a bigger form also, but just to show one point, I am drawing over here anther separately. This is how beta actually anther is. There I have drawn it like this now, but actually if I want to show a microscopic diagram of anther only, this is how it is going to appear. How? These are the 2 lobes, this is 1 lobe and this is 2nd lobe. Both the lobes are connected with the help of connective, so in the center there is connective. Correct? Now each lobe is having 2 chambers, so these 2 chambers of this lobe and these 2 thecas of this lobe. This is how anther appears to be. Now how many sides are there? Can you tell me how many sides of the anther can you see? A side, B side, C side, D side. So can't we say that our anther is tetragonal? Yes or no? 4 sides are there in the anther? Yes. So next point which you are going to write for anther is that anther are tetragonal. Right? 4 sides. Plus how many total thecas are there? 4 theca. 2 in one lobe, 2 in another lobe. So there are 4 chambers. Hence you can say that anther is also tetrasporangiate. Right? These are the sacs in which pollen grains are produced. So there are 4 chambers, 4 thecas in total anther, which you can say they are also tetrasporangiate. Each sac is also known as sporangium. So it can also be known as tetrasporangiate. Understood everybody? So these are the points which you have to remember for anther, that anther is a site of pollen grain formation, which is a male gamete carrying body. It is bilobed. Each lobe is having 2 chambers, so it is dithecous, tetragonal and tetrasporangiate. Now moving ahead with the third structure, filament you understood, anther you understood. Now the third part of a typical stamen is connective. What is connective? It is a sterile tissue. It is a sterile band that holds the 2 lobes of anther together. This much is only there for connective. It is a sterile tissue. Connective represents sterile tissue which holds the 2 lobes of anther together. Right? Yes, ma'am. Very good, students. Okay, now structure of stamen is clear. You have already come to know that where in stamen actually pollen grains are going to be produced. Now our main aim is to understand that how pollen grains are produced. So for this we are going to take the structure of anther more in detail. Okay. Sorry, I think, yeah, sorry, this is just an extra page by chance. Coming to the main topic, that is microsporogenesis, the mechanism of formation of pollen grain in the anther. That formation, that process is known as microsporogenesis. So now we are going to study microsporogenesis. Let me first write the definition. What is microsporogenesis? Microsporogenesis is simply formation of haploid microspores Microspores are actually your pollen grains, right? From diploid mother cell. From diploid mother cell. Where? In the anther. So where this big process is taking place? Inside the lobes of anther. Correct? Chalo, now let's try to understand that how microsporogenesis takes place in the anther. What is the mechanism? So for that, again you have to understand the detailed structure of anther. Beta, when anther is young, what I said, when anther is young, it is just represented by a layer of epidermis that encloses lot amount of parenchymatous cells called sporogenous tissues. So suppose this is my young anther, which is tetragonal, tetrasporangiate with 4 sides, correct? Held together with the help of connective. Now this is young anther. So beta, when anther is young, it is simply surrounded by a single layer, outermost layer, which is called as epidermis. So this is whole epidermis. I'm not going to draw the complete. Yes, you'll understand here also there is single layer of epidermis. This epidermis inside encloses large number of living cells, parenchymatous cells, thin-walled cells called sporogenous tissues. Right? This is how the young anther appears to be. Now, when this young anther matures, then what happens? One of the cell of sporogenous tissue, one of the parenchymatous sporogenous cell below the epidermis starts developing, starts growing in size. Right? Now what will happen? Let's see again. Below the epidermis, correct? One of the cell of sporogenous tissue will grow in size. And this grown cell of sporogenous tissue below epidermis is known as archaesporal cell. It is known as archaesporal cell. So when anther tries to mature, first step is formation of archaesporal. Chalo, read it till here. First of all, young anther consists of epidermis which surrounds parenchymatous cells called sporogenous cells. Next, one of the sporogenous tissue cell gets mature to form archaesporal. One more point beta you can remember. Formation of archaesporal cell is hypodermal. Hypodermal means below epidermis. So archaesporal cell is hypodermal in origin. Very, very important line. NCERT has written this. Question will come on this line. Hypodermal in origin. Right? So this archaesporal cell is produced, which is hypodermal in origin. Now beta, this archaesporal cell, wait, let me adjust one page. So yes, now this archaesporal cell, my dear students, will undergo, now let me take out this archaesporal cell, okay. This is going to undergo a transverse division called periclinal division. So it is going to undergo mitosis, a transverse mitosis, to produce from one round of mitosis. How many daughter cells will be obtained? 2. So from this, 2 daughter cells will be obtained. Upper one is known as parietal cell and this lower is known as sporogenous cell. Correct? Understood? So what is the scenario? Scenario is this beta. This was my young anther having single layer of epidermis plus lot amount of parenchymatous cells, because of which the one of the parenchymatous cells grew in size, which is known as archaesporia. So this is the condition. Now you're having two cells, upper parietal and lower sporogenous. Now this upper parietal further undergoes repeated mitosis. Lot many mitosis takes place in this upper parietal cell to produce large number of cells. Those large number of cells then differentiate to form remaining three wall layers of anther, that is endothecium, middle layers and tapetum. Once again I am repeating, please listen to me carefully. The upper parietal cell, then it undergoes mitosis, repeated mitosis to produce remaining three wall layers of anther. One wall layer is already there, which is epidermis. See, epidermis is already there. This is the anther wall layer. But a mature anther has four wall layered. So remaining three are produced by parietal cell. Correct? That means the young anther is having only one layer as epidermis. However, when my anther will mature, when anther is going to mature, it will have how many wall layers? Four wall layer. One already existing, three remaining are produced later on. Chalo dekho bhai. See. So this is my already existing epidermis. Below epidermis, with the help of parietal cell, will be produced endothecium. So outermost is epidermis. Below epidermis comes endothecium, which is also single layered. Below endothecium will come 2 to 3 middle layers. Right? So these are 2 to 3. This is not single layered like epidermis or endothecium. Rather, middle layers are 2 to 3 layered middle layer. And the innermost layer which will be produced is known as tapetum. This is very, very important layer. Although all layers are important, but tapetum has its own significance. So this is how mature anther will look like. It will have 4 wall layered anther. Getting my point? Yes or no? So this is your mature anther. Now this tapetum will be surrounding large number of sporogenous cells. You know, these sporogenous cells. Apart from other parenchymatous cells, these sporogenous cells will also be enclosed within your tapetum. Understood everybody? Till here? Yes, very good. I'm just leaving this sporogenous cells, these orange colored sporogenous cells over here. We'll discuss them. We'll discuss about them later. But first try to understand the functions and characteristic properties of all the 4 wall layers of anther. Chalo. So first let's start with epidermis. Beta, epidermis, it is the outermost layer. Being outermost, its most important function is protection. It is already present. It is not produced by parietal cell. Fourth, in case of Androecium, but Androecium is a parasitic plant. In them, special property has been observed that in Androecium, the epidermal cells, they develop some fibrous thickening. In them, it is known as exothecium. Correct? 4 points understood? Chalo, likho bhai, write it. So epidermis, first of all, it is outermost. Second, it is protective in nature. Third, already present in young anther, right? And fourth and very important point you will write, in some plants like, for example, Androecium, it is a parasitic plant. In Androecium, epidermal cells develop some fibrous thickenings. This deposition of fibrous material have fibrous thickenings. So there, thus in Androecium, it is known as, yes, Endothecium. So yes, understood till here? What is epidermis? Yes or no? Very good. Okay, so I hope Endothecium is clear to everybody. Now, after epidermis, inner to epidermis lies the next layer, which is known as Endothecium. Endothecium is the layer, beta, which develops from parietal cell. It lies inner to epidermis. The cells of endothecium, they are hygroscopic in nature. That means they have the tendency to absorb moisture. They have the tendency to absorb water from the environment if outside conditions are wet, and vice versa. That means if outside conditions are dry, they tend to lose their moisture and then they start contracting, right? So these cells of endothecium, they are hygroscopic in nature. So first point that you're going to write for endothecium is that they lie inner to epidermis. Second point, develops from parietal cell towards the maturity of anther, right? Third point, you are going to say the cells are hygroscopic in nature. As a result, fourth point, they help in dehiscence of anther. Now what happens, beta? See, towards the now inside this, the pollen grains are produced. Now once pollen grains are produced, they need to come out from the anther. For this, anther need to burst. So bursting, dehiscence of anther is promoted by endothecium. How? When the outside conditions are dry, the cells of epidermis, they tend to lose their moisture, because of which the cell starts contracting. Due to the contraction of the cell, a pressure is applied at this point, at this junction, which is known as stomium. So kind of pressure develops because of the squeezing of the cells, because of the contracting of the cell. As a result, from the stomium, anther burst, releasing all the pollen grains in the air. Correct? Understood this point? So dehiscence of anther, bursting of anther is a characteristic feature of endothecium. Inner to endothecium lies 2 to 3 layered parenchymatous middle layers, which again develop from parietal cells, as well as they are ephemeral in nature. That means their lifespan is very, very short, and as such they do not have any other specific function or role to play in the anther wall layers. Simply you can say that okay, they can help endothecium in bursting, but specifically middle layers do not have any assigned function. They are very ephemeral. Their lifespan is very, very short. They soon get degenerated. So next point, what are you going to write for middle layers? Number 1 point, first, that they are present below endothecium, right? Second point, they are 2 to 3 layered. And third point, that they are ephemeral. Ephemeral means they degenerate soon. Their lifespan is very, very short. Right? Understood till here, everyone? The 3 layers of anther Now talking about the fourth and the innermost layer, bachcho, which is tapetum. Tapetum is the innermost nutritive layer that develops from upper parietal cell. Correct? Now let's write few important points of tapetum and then we will discuss functions of tapetum. First point, it is innermost layer. Second point, develops from parietal cell. Third point, beta, it is nutritive. Means it provides nutrition to the developing pollen grains. Now you know, see this diagram. Now tapetum is just nearest to sporogenous cells. Tapetum layer surrounds the sporogenous layer. These sporogenous cells, you know, they are only going to later develop into pollen grains. Now for the development of pollen grains, nutrition is required. Pollen grains, while they are developing, they need nutrients. So that function of providing nutrients to the developing pollen grain is the role of tapetum. So tapetum is nutritive in nature. It provides nourishment to the developing pollen grains. So next point you are going to write is they provide nourishment to the developing pollen grains below it. As next point, they surround. Tapetum surrounds the sporogenous cells. The parenchymatous sporogenous cells. Right? Very good. Now, beta, the cells of tapetum, they show polyploid condition. They have multinucleate condition, because of which they have high DNA content. So 3 points what I told you. First, the tapetal cells, they have polyploid condition, means normally anther is diploid structure, right? The ploidy of anther is 2n. However, in the tapetal cells of anther, more than 2n chromosomes are found. So polyploidy is the thing which is reported in tapetum. So next point, the cells of tapetum, they show polyploidy. Next, they are multinucleate. That means if karyokinesis takes place, sometimes in tapetum followed by karyokinesis, there is no cytokinesis, resulting in multinucleate condition, due to which per cell tapetum, they have high DNA content. So I hope will you remember all these points for tapetum. First, they are innermost layer which develops from parietal cell. They are nutritive in nature. They surround the lower sporogenous cells. They show polyploid condition, multinucleate condition. As a result, they have high DNA content inside them. Correct? Now, on the basis of function, tapetum is responsible for providing nourishment to the pollen grains, yes or no, which are developing below them. So depending upon how the mode of providing nutrition, tapetum can be of two types, secretory tapetum and amoeboid tapetum. So depending upon the mode of nutrition, bachcho, tapetum are of two types, that is secretory and second is amoeboid tapetum. Bacha, what is secretory and what is amoeboid tapetum? Secretory means when tapetum cells, they remain in their position. Below these are the sporogenous cells which will develop into pollen grain later on. Correct? And these are your tapetal cells. Correct? Now secretory tapetum means the cells will remain in their position. They are not going to detach from their position, and from there only they will secrete the chemicals, the nutrients, which will be then received by these sporogenous cells, right? These secretory tapetum cells, they secrete chemicals like pollen kits, compatibility proteins, some enzymes like callose enzymes. So all the secretion is done by the tapetal cells, not leaving their position. So what will you write in secretory, beta? Tapetal cells remain in their position, remains in their place and secretes the nutrients. And what are those nutrients? Like, for example, pollen kit, right? Some proteins called compatibility proteins. Some enzymes like callase enzymes, etc. Right? Contrary to secretory are the amoeboid tapetal cells. These are primitive tapetum cells, which beta, they leave their position, they degenerate from their position. Wait. Let me use some other color pen. So these are the sporogenous cells, red one. Now some tapetal cells will come out from their position and then will sit on the sporogenous cells and give them nutrition. So can you see the difference? In amoeboid tapetum, the tapetal cells, they come out from the position, they directly come and settle on the sporogenous cells to provide them nutrition. However, in secretory, they are not getting detached from their position. So what will you write over here? Tapetal cells, when detaches from their position and then provide nourishment to the sporogenous cell. Clear to everybody? What is tapetum and types of tapetum on the basis of their method of providing nutrition? Very good. Now, moving ahead, beta. Parietal cell, its property is done. Now, if you remember, I told you, leave lower sporogenous cell over there. Later, we are going to talk. Now, we are going to see that how the lower sporogenous cell, which developed from archaesporal. You know, archaesporal produces upper parietal and lower sporogenous. So, function of upper parietal you have already seen. The three remaining wall layers and their properties and their functions. Now see that how this lower sporogenous cell functions. So now this is your lower sporogenous cell, which undergoes first repeated mitosis to produce more number of diploid microspore mother cell, right? So this sporogenous cells actually divide by mitosis to produce more number of diploid cells, now called as microspore mother cell, because now these microspore mother cells will now undergo meiosis, reduction division, to produce haploid microspore. So here in Anther, actually, with the help of sporogenous cell, pollen grains are produced. Now these microspore mother cells, beta, will undergo meiosis, will undergo reduction division to form haploid microspores. From each microspore mother cell, 4 haploid microspores are produced. When one microspore mother cell will undergo meiosis, they are going to produce 4 4 haploid microspores. These microspores are actually pollen grains, and all four are functional. These all 4 are beta functional. None of them get non-functional or degenerate. All the 4 microspore haploid cells produced by the meiosis in microspore mother cell has the capacity to act as pollen grain, the male gamete carrying body, the male gametophyte. And this phenomena is known as, this whole phenomena is known as Microsporogenesis. So today you have seen that how within the anther, within Androecium there is anther, and within anther that how pollen grains are produced with the help of microsporogenesis. Is this concept clear to everybody? Now just try to recall everything. There was an anther, a young anther having epidermis and some parenchymatous cells. One of the parenchymatous cells matures to form archisporial that undergoes division to produce 2 cells, upper parietal, lower sporogenous. Upper parietal beta undergoes repeated mitosis to produce the remaining wall layers of anther, that is endothecium, middle layers and tapetum. Coming to lower sporogenous, this lower sporogenous cell in anther is actually responsible for producing pollen grain. Now these pollen grains, now endothecium is going to burst, and now pollen grains, the male gametophyte, will be released. Clear to everybody? Yes or no? If yes, then for today we are going to keep it till here. Structure of pollen grain and where are the male gametes. Right now you have only produced pollen grain but not male gametes. So formation of pollen grain is done, but structure of pollen grain and within pollen grain where are the male gametes, that topic we are going to take in our next class. So I hope till here things are very much understood by all my dear lovely students. As you can see, there is a homework on the screen. Question number 1. This is homework for you all. Please write the answer in the comment section. I have seen many of you are writing the answer, but I expect that all of you should do it. Please, if you're studying, then it should be done in a proper manner. Try to write if you're listening to the lecture carefully and properly, then I don't think answering the question will be a very big deal, right? Whatever question I write in the last is actually taken from the lecture itself, just to ensure that you are listening to the lecture carefully. So this is your second homework. Please write the answer in my comment section, and till then I will say thank you. See you in the next class. Bye bye everyone. Take care. Keep studying. Keep working hard.